So it's a great pleasure to be able to talk to Sydney Brenner, who I've known for over 30 years now in King's College. Thank you, Sydney, for doing this interview. Could you tell me first where and when you were born?
I was born in Germiston, which is a small town outside Johannesburg in South Africa, and I was born on the 13th of January 1927.
And firstly, something about your parents and their background would be very interesting.
Well, and my father came out from Lithuania as an immigrant to South Africa. Went to South Africa because the fare was cheaper, and he had a brother there, and that we reconstructed to be about 1911. My mother came from Latvia, at least on the border of Latvia and Russia, and she came out in 1922, so she lived in Russia during the war, the revolution, and that is what I remember from my youth, being regaled with stories about white Latvians and red Latvians and white gods and red gods. And my father was a shoe repairer, and we lived initially in rooms at the back of his shop, so I grew up with the smell of leather, and that was my early life there.
Tell me something about their characters and how they influenced you.
Well, my mother was someone who had, I think, great ambitions for her children. My father was illiterate. He couldn't read or write, but he had a great gift with languages. He spoke several languages, including an African language. My father was he did that, but I think the important thing is that my mother encouraged me to read. She knew how to read, and I learned to read because I visited another old lady who was a friend of my family, again another immigrant from Russia, and learned to read from the newspapers that were spread out on the table in lieu of tablecloths.
And then what was your first school? Do you remember your first school?
Yeah, I went to a kindergarten because a lady who actually ran it was attached to the Presbyterian church in there. Her name was Miss Walkinshaw, and she ran the kindergarten, and she came in as a customer to my father, and she found me at age 4 and a bit reading a newspaper on the floor, and so she said my father should send me to her kindergarten. And of course my father said that they couldn't afford to, but she took me in there, and I did the first 3 years of primary school, which was in South Africa was grade 1, grade 2 and standard 1. I did that all in a year, so that when I went to the government primary school, I went straight into standard 2 at the age of 6. So I managed to compress 3 years in 1 year, and so I was always about 2 years ahead, younger than everybody in the same class, which was not helpful.
Were you quite small at that time?
I was small at that time, and a bit of a coward, I say, but often got beaten up. It was that kind of school, yeah.
And then after that school, where did you go on to
Well,
school?
then I went to a high school called Germiston High School, and at the end of that I matriculated when I was just less than 15, but I'd won a scholarship, a bursary to the university, given by the local town council, which meant that I could go to the university in Johannesburg to study medicine. Of course, by that time had a very big interest in science, a major interest in doing that, but medicine was what was being financed. And so that's how I got into biology, if you like, although I had a lab of my own. That is, I had advanced education in garage chemistry.
Garage?
Garage chemistry.
Garage chemistry,
That is, doing it in a garage, yes.
Were there any teachers at that high school who particularly influenced you?
I can't remember any of them that influenced me. In fact, I was completely astounded by their ignorance because I received most of my education from the Germiston public library, which was the endowment of Carnegie, and spent nearly all my time reading there.
You mentioned that you were already interested in science. How would that happen? Just curiosity, or?
Well, I think it is, I think it is, most children are interested in nature. And of course one of my ambitions was to find out about nature. So the way to do it is, of course, to take it apart. I remember a few years ago I was in Japan and several elderly gentlemen were discussing how they became interested in science, and one of the physicists said he got his interest in science by taking a crystal set apart and putting it back together again. And I said, well, I did the same thing because I used to take flies apart, but I couldn't put them back together again. So that became one of my interests. How would you put it back together again?
So at the age of 15, 16, you were ready to go to...
When I was 15, I started.
At university?
At university.
You must have been by far the youngest.
Yes, I was 2 years younger, at least 2 years younger than most of the people there.
Which university was it?
There's one in Johannesburg, University of Witwatersrand. And I lived at home, I travelled every day, first on a bicycle about 2.5 miles to the station, then on a train, and then walking to the university from the railway station, and taking sandwiches for lunch, and going in the morning. We had a much tougher regime there. We had lectures or laboratory sessions every day and Saturday morning. Everything started at 8 o'clock there in the morning. So it was quite wondrous to have to set out at about 6.
Did you enjoy it?
Yes, because I think there I actually did meet people who were doing things. There was a man in the botany department who I discovered just in walking past, and he let me come and work in his lab, who was doing chromatography, which I thought was astounding, separating all these colours from plants. So that I did enjoy, and I learnt, of course, we did 4 subjects: botany, zoology, chemistry and physics. I liked the zoology, but of course we had to learn a lot of things there in order to write examinations in vast details, you know, all the embryology of things and so on. And so after the first year, I then moved to the medical school, where I did anatomy and physiology, and that was interesting as well. And then it had been discovered that I couldn't qualify because I would have been under 21 when I finished medicine. And so I was able to take a year out to do a Bachelor of Science degree in anatomy and physiology. And that became heaven, because there one worked in a lab, and I took out 3 years. I did a BSc, then a BSc Honours, and then a Master of Science. But then I think I was already doing scientific research. And I realized then that I wasn't a good medical student. In fact, the other day I gave a talk entitled "The Worst Medical Student of 1950," which, but in fact, at the end, although I resented, having to go and complete it, and I should say, I completed it because I'd finished all these other science things by the time the war ended. And everything had opened up by then, and people had started to go abroad again and so on. And the question was, I thought, since many of my friends were going, what should I do when I finish my master's degree? And should I go abroad and try to do advanced work? But I was advised to never get a job as a scientist. And the only jobs for my interests, which then would be in medical schools, and you had to have a medical degree, otherwise you were a second-class citizen. So I stayed back to finish 4 more years of medicine, from 1947 to the end of 1950, and of course then I did go abroad in 1952.
So how long had you been altogether at that university?
I was at that university from 1942, I think almost 9 years. Where I'd done a medical degree. I landed up there actually teaching. I was a lecturer. In fact, I became a sort of lecturer while I was still a medical student. I used to have to go and teach people physiology. But that was good because, when you teach a subject, you have to really know it and understand it. And I became a great expert on one of the things which nobody could understand, which is, calorie intake and all of this. Now become very important. So I knew exactly why you burnt all the things at the rate you did and didn't at the rate you didn't.
Well, were there by this time any teachers or colleagues who you particularly remember as influencing you at university?
Yes, well, there were 2 people there. The one is, there were many people there that influenced me. The most important one, I think, in that time was Raymond Dart, who was the professor of anatomy, but more so was a man called Joseph Gilman, who was then a lecturer in histology in the anatomy department, later became professor of physiology. And he was the one who encouraged everybody to read widely, but of course didn't like to be contradicted. And so many arguments became quite heated after a while. But I think discussing scientific problems And actually working in the laboratory was a tremendous experience, and that I think, and also doing things right from the start, because there was nothing there. I wanted to do an experiment which required an amino acid, but of course no amino acids had reached South Africa since the beginning of the war. And so I prepared the amino acid. I found a book, I read it, I went to the barber shop, I collected all the hair, and I cleaned it, of course, and then hydrolyzed it and made cysteine. In fact, I got so good at that, I was making all the amino acids from various things like milk, powder, hair dropping, well, hair cuttings for nutrition experiments.
It's rather like Newton grinding his own lenses.
Yeah, well, I think this is what we did. I even built an ultracentrifuge and used it in what I considered to be one of my really clever experiments.
Were your parents happy that you were spending so much time at
I think
education?
they were very generous in the sense that, of course, in classic Jewish families, learning is highly regarded. But my mother would have been much happier to see me at that time as, let us say, a specialist doctor, because that had very high social status, you know, with a nice car and a good practice and so on. And I think that I think it was only very late in life she realized that I'd got somewhere after all.
So now you move on from there to postgraduate?
Well, yes. Well, what happened was, I think I know what I was interested in, but the subject hadn't been invented. It's called molecular biology. Waddington, that's Caroline Humphrey's father,
Yes.
came out to South Africa at the time, and he encouraged me to go to apply to Cambridge, which I did, and of course they never even replied to my letter, so that wrote Cambridge off. But I won a very rare, there was only one per year for the whole of South Africa. It was the scholarship of the 1851 exhibition. So I won it for that year, 1950. I applied for it. And where it came to go is, where it came to for me to decide where to go, I asked the advice of the principal of my university, who was an old Oxford man, and he recommended me to go to work with Cyril Hinshelwood.
Cyril?
Hinshelwood.
Hinshelwood, yes.
Hinshelwood was the professor of physical chemistry and had written a book called The Chemical Kinetics of the Bacterial Cell, which sort of fused physical chemistry and biology. And so I applied to him and got accepted. And so I then went in 1952 to Oxford to do a DPhil in physical chemistry.
Was it something of a shock to arrive at Oxford?
Well, I'll tell you, you know, we had a group of people in South Africa, because apart from doing all the science, I was with a very talented group of people, some of whom are still alive, where we actually made films. And we decided, for example, to make a film of one of, I think it is I think it was by James Joyce, but I'll need to try and recall it. However, but it was about England, and it was about Wales.
Yes.
So it wasn't but James Joyce. I think it was Dylan Thomas,
Yes.
actually.
Mm.
And so, of course, we had to imagine
Mm.
what England was like without ever having seen it.
Mm.
And however, we had this, of course, everybody was connected with England just by books and by the sort of education we'd had in the school. But it was a shock when I actually came there, because the one thing that I found almost desperate was food rationing was the law. And for 2 years I just dreamt of food, because one had very little. However, after a term in Oxford, May and I got married. She was in London doing a PhD, and so we got married and we set up a house in Oxford when we both finished our PhDs in 2 years at the time.
Gosh.
And I then won a scholarship, a travelling scholarship from the Carnegie Corporation to go to America for 4 months. In the meantime, as I've said many times, in about November 1952, I had a very good friend in Oxford called Jack Donitz, was a crystallographer and in contact with all the major figures in the field. And he told me, I came to Oxford with two half-wrong theories. I mean, it's important to measure the amount of wrongness in a theory, because even a tiny amount really kills it. So I came to England with the idea I could determine the structure of DNA by some method, but it was all half-baked.
It was already a well-known problem.
No, it wasn't a well-known problem. It was a rather fringe problem, because, I mean, genetics had its problems, but to connect it with this chemical, because it wasn't actually accepted that the genes were built out of DNA until much further on.
Yes.
And people fought a dying battle. That's simply because in the field, most people felt genes do complicated things, so they better be built out of something quite complicated, like a protein, you see. And of course, J.B.S. Haldane had coined the concept, I think, as early as 1929. It's the greatest misleading concept in our science, in which he characterized genes as autosynthetic enzymes, which is wrong. And so all of this, just I said that this, but of course I didn't have that. But of course I had become interested in it. And my interest was really taken up when, of course, I went to Cambridge. After hearing about Watson and Crick, we actually went in April of 1953, just before the paper was published. 3 of us, Jack, Leslie Orgel, and myself, went to Cambridge and met Watson and Crick and the model. And of course, that was a revelation. And it was, one saw the light, and since then one never turned back.
They had already discovered?
They had already discovered the structure. And we saw the structure, and its implications were just blindingly, I won't say obvious, but blindingly clear. And you sort of, after 5 minutes, you sort of realize, well, there's the coding problem, there's the copying problem, so we better get down and solve all of these. And that's essentially was the real turning point in what one was going to do.
What was your first impression of those famous two people? Because you worked a lot with Crick later, of course. But
yeah, well, Francis, I mean, for that one day, of course, Francis wouldn't stop talking, basically. But Jim just always gave me the impression then, and I wrote about it later, of a kind of irritated bird. You know, pecking this, pecking that. But the impression was they'd made a breakthrough. You know, this was what the world needed, although no notice of it was taken at all.
really,
For quite a time, yeah. It was the most amazing thing, because everybody sort of doubted, A, the way it was produced, B, there were problems there, but to me it was just very clear this is what we have to get on with. And so for quite a time, the field was essentially occupied by a tiny band of people. You could 20, 30 people who saw that this had reformulated major questions in biology. And although one always writes these things with hindsight, it became clear that the sort of problem that most people were preoccupied with in biological systems at the time were problems of matter, which is how do you make all these various, you know, compounds that you find in the body, B, where you get the energy for them. But no one had said, look, the important thing is how to make these proteins, how to get the amino acids in the order. This is the coding problem. And we knew that that was the problem, and we knew it only from the work of Sanger. So while I was at Oxford, there was a club there called the Alembic Club of Chemists, and Fred Sanger came to talk, I think it was in 1953. Yeah, that's correct. And he came to talk there because he had just assembled insulin, made the sequence of insulin. And he had wonderful gadgets for his lecture, which consisted of little blocks that as he made the pieces, he turned them around and you could read the amino acids on the little block, you see. And it was a great thing. But I'll never forget that after his lecture, Robert Robinson, who was the doyen of British chemists, stood up and said that this was remarkable, because Sanger has proven proteins actually have a chemical structure. No one believed in it. They were some kind of gloop or glob which had just polymerized together. No one had any idea that they had a defined structure in the form of the sequence. And that's the thing that matters. And once we had that, then you have to ask yourself, how do you get these things in a row at the same time? Now, I'd also had a background. I had a very good friend in South Africa, who was a mathematician, called Seymour Papert. And I got very interested in certain aspects of this. And in particular, I did read a very interesting article by a man called John von Neumann. And I still have the book, which was written before the Watson-Crick. It was how you might build an autosynthetic machine, a machine that could build itself. And of course the logic of this is exactly what was embedded in the DNA. But of course nobody knew that at the time, so I'm sure that if someone is just blatantly writing the history of ideas, he could actually look at Van Neumann's publication and say, my God, there he's written about DNA. And there's Watson and Crick, influenced by Van Neumann. But in fact, the most amazing thing is that in 1949, it is, Pauling and Delbruck had written a paper about complementary replication, which is I make you, you make me, and we've got a double helix if you like. But they'd forgotten about this. It's something I always tell students, you can read about ideas, but it's how seriously the proposers take them that counts. If they are just thrown in, or we can get a machine that makes random ideas, but you can't lay claim to them unless you are a supporter of them.
You mentioned Fred Sanger, and I'm hoping to talk to him.
Yeah, good.
Can you say something about him? He always struck me, he's at King's of course, as a very modest, quiet man.
Well, he was a unique scientist, Fred, and I've always been impressed. There's no one else like him, in the sense that he saw that, and as he did first in protein chemistry and then later on in the whole of DNA chemistry, that determining the sequence, determining how the components are arranged is important. And he devised what were very simple techniques in order to achieve this, and carried them out. And he liked to work in the lab. I mean, when he retired, he put down his pipette and said, that's it, and walked out. But I think he was a real scientist, and I think the things he had major impact. But the one thing about Fred, which is very amusing, was I noticed that he went up by factors of 3 when he started to sequence. He first did something which, let us say, was 6,000 bases long. He first did something in the RNA field, which was very small, and then he got up to 100, you see. Later on, he kept on going. He got up as far as 50,000 this way. And I had a colleague, and I had an idea about how we might purify a product of a gene. And I told this young man from America who was working with me at the time that we should go out and talk to Fred to see if he'd be interested. And I said to him, when he asks you how big is it, say it's 300. So, but he chickened out at the end because he thought this was too big. So Fred said, how big is it? How big do you think it is? And he said 100. So Fred said, oh, we're not interested, we've done 100 already. So that was...
Fascinating. One other question about DNA. I've heard sometimes that, and it's mentioned in the double helix, that I think he was a Trinity originally, and then later a King's mathematician, John Griffiths, who you knew.
John, yes.
He had an important role in...
Well, yes. John was... I knew John very well, and John was a very good mathematician, and he is... he's famous for 2 things. Oh, well, famous. Nobody's famous who hasn't done work over the last 2 years because everybody else there belongs to the common epoch, which is the one preceding the last 2 years. And he solved or he helped to solve, there's a very famous paper by Crick, Griffiths, and Ogle. It's called the Cumulus Code. And that was, if you took 20, if you took four things and they wanted to code for 20 amino acids, how could you arrange them so they made other conditions? And one of the conditions was that you should be able to start anywhere and it would still make sense. Now, of course, taking 3 bases for each codon gives you 64. And that's too many for the amino acids. But if you make that code commaless, which you can do very easily because it says if the sequence, just to give you one example, if the sequence consists of the same letter, the triplet, like AAA, you can't make that commaless because you wouldn't know where to start if you had 2 together. So there'd be several ways of reading this. And so that eliminates 4 out of the 64, leaving you with 60, which happen to divide into 20 classes, each containing 3, and that was the commaless code. Marvelous, except it's wrong. And that's the great thing, that you should always suspect that an elegant solution is probably not nature's solution. Because as I often say, mathematics may be the art of perfection, but biology is the art of the satisfactory.
Although elegance is a nature, your famous nematode worm was
called C. elegans.
Yes.
I didn't give it the name. It was given to it by a Frenchman who discovered it, yes.
Okay, well, let's go back to your life. You've done your PhD and you went to America.
I went to America and then I went for 4 months and did a lot of work there. And then, but in the meantime, I started to discuss with Francis about coming back to England, in fact, joining him in the MRC unit. And so I went back to South Africa because I had to obey certain of the conditions of the fellowships I had. And 2 years later, after arrangements had been made, I came to Cambridge at the end of 1956 to a 3-year job at 1,100 a year with 3 children. So, but I came to do, and I think that was the really beginning of what was an incredibly exciting time in science.
Working with Crick, you mentioned various things, for instance his famous notice on the wall, which I have on the wall in my office now, I'm reading rots the mind.
Yeah.
But you said he was actually a rather well-read.
Yes, well, this is what I've written, a little piece on this, you see. I didn't know where he got this from. But he turned up one day and he put it up on the wall, reading rots the mind. I later learned that he got it from Christopher Longueville-Higgins, who was a mathematical physicist and professor of theoretical chemistry. Because Christopher really believed there were only 2 things you could think, or you could play the piano, basically. But reading rots the mind. So Francis had this because of the things, but of course it was hilarious because he would sit underneath this reading all day. And when he went to the Salk Institute in the mid-70s, the sign was still there above his desk, but the entire room was full of books on the brain, through which he was steadily reading. So we have to
guess what he meant.
We have to guess what that was.
But what struck me about your account of your relationship is these conversations.
Yes.
Working things out in words in conversation.
Well, I think I think that was a very unique combination, because the rule was you could say anything, I mean, provided it wasn't stupid. And in many cases, one would get it half wrong. You see, someone like John Griffiths will go away and think about it and then come and produce the finished version. I can't think that way very easily. I have to churn it around, and I churn it best within conversation. And I think we produced many things that way, and even up to almost the last week I spent, I saw Francis in there, and we spent hours discussing, the brain and how one would actually do things, and so that's, as I think, a very productive relationship, because I think we've only written about 3 papers together, although we were colleagues in the same thing, and there's only 1 paper which is just by the 2 of us. It was just a short thing, but it was pretty nice to have done it.
Did you, after you'd have those conversations, would you rush off and write ideas down from the conversations, or...?
No, Francis would be the one. I would actually go and try and see if I could do an experiment to pin down those ideas. And that's been my habit ever since, which is ideas are fairly cheap, and they circulate, so to speak, in some space of their own. And the thing that really clinches them is the experiment which proves they're right. And of course, this idea that you can prove something correct by science is now sadly disappearing. Everybody thinks their task is to describe nature in ever increasing minutiae.
You say a number of times that it's very important to just have lots of guesses, many of which are wrong and some of them are half right. I was very struck, one of my favourite quotations from Einstein is, unless an idea starts off as absurd, there is no chance for it.
Yeah, correct, you see, because most ideas are wrong in the beginning. I mean, you know what you want to try and get, but in fact you've started on a slightly wrong path, and you've got to a thing and you've thought of, very triumphant till you've thought about it and you discover there's a little snag in it, and so you must reiterate it. And so I think many things go, and I think a talking exchange and being able to tap evidence from all sources, which one could do with a number of the people that I'd had contact in my life, is I think very valuable, because this is an interesting way of generating a cooperation, a cooperative thing, which is usually produces something that's quite interesting, I mean, and often right.
Well, you make a distinction between correct theories and true theories.
Yes, you see, the Commonwealth Code is correct, but it's not true. That it's logically correct. Many theories are logically wrong, many can be wrong because there's some fatal flaw either in the deductive or whatever it is. But the one that coincides with nature is what we're interested in. And we want to know how nature does it. And therefore, it's true, you could probably do it better than nature, but that's not the name of the game.
Coming back to your conversations with Crick, did you develop a sort of, not exactly secret language, but a shorthand
Well,
Because
after a while, you see, well, we got one of the things that, I've invented a story about Francis, which is he would sit at his desk and ponder on one of the problems, and then would say something. Now, he was once talking about the imaginal discs of flies and how they might develop, and then when he got frustrated, he quite often banged the desk and said, God knows how these imaginal discs work.
Mm.
So from that I created a story of Francis going to heaven, so on, and then finally meeting with God and saying, how do these imaginal discs work? But of course God then says, we don't know. But we've been building flies here for 200 million years and have had no complaints. So I think, but that is how it is. And then, I think you have to be in possession of tremendous amount of the detail of the subject, and also know a lot of things outside of it, because sometimes there's an indication from one experiment which has no reference to this, but suddenly you realize, boy, I can explain that, and that becomes the interesting thing to do. So, the shorthand was effectively the jargon that had been developed about this corner of biology. And of course, if other people came into this, I wouldn't know what you were talking about, because there were a whole lot of words which began as lab jargon, some of which actually became part of the language. And I think that is, I think one of the things that's not realized about science, and at least, is that I think it works more like a medieval guild, in which you have, an experienced journeyman with his apprentices, and they learn from the master, so to speak. And nowadays, many people think you can learn it by taking, 49 courses and studying there, and you can do it all by some kind of teaching exercise. But just sort of hanging around and talking and discussing things is, I think, the best way of learning science. Just being there and listening to and of course that's why in the old LMB, the tea room was the open thing, and you would talk to people in other fields and learn things. And Francis was a great learner. He always wanted to learn new things, because he thinks they would throw light on us.
one of the reasons I was asking about this sort of special language was to lead out to ask you to describe that famous meeting in your room in King's, where only he seemed to understand what you were talking about.
Well, you see, we were talking there about an experiment. And we were talking about how does information get out of the dream. And in those days you could say there's several possibilities. One, it doesn't get out of the gene, it's used directly on the gene. Okay. Two, it gets out of the gene in this way and in that way. But we were faced with an incredible paradox at the time which defied solution. And that was the following, if I can just put it in this way. The paradox was that the little gadgets that made proteins in our cells, which are called ribosomes, were full of RNA. And I always believed that RNA was an intermediate. But the paradox was that the composition of the RNA was constant, independent of the composition of the DNA. So the DNA in bacteria could vary over a factor of 10 in the relation between the bases. Of course, being DNA, G always equals C and A equal T, so it was the ratio of GC to AT that could change from 1 in 3 to 3 to 1. And that's a paradox. And there were all kinds of solutions to it. And at that experiment, I suddenly realized that if there was a copy of the DNA which traveled to the ribosomes, because we held on to the fact that the ribosomes were the vehicles and that RNA was something, but it couldn't be tolerated, then it should have the base composition of the DNA. And I suddenly remembered that there was such a minor component described by other people in another experiment. This is the thing. It was described by two people called Volkin and Ostrakhan, and many people thought that that was actually used as a precursor for the DNA. But suddenly putting those facts together with that in one blinding sort of boom, you know. And I suddenly began to say, I said, the Volkin Ostrakhan did. Most other people didn't know what we were talking. Francis got it immediately. And I said, that carries the message. And by that evening, I had formulated the experiment to test it. And Francis had already written a paper about it that evening, but he was dissuaded from publishing it. I think we should have published it straight away. There's the concept of this, with the evidence already in the literature. And as it is, it waited for an experiment, but it was good that the experiment worked, because nobody believed it, basically. But again, I think had one said that, everybody would have poo-pooed the idea. But then it explained hundreds of facts, and actually had a physical existence. So that was very exciting. And that was, then, you know, there followed quite a long period till about November of that year to try to actually prove that it existed.
What was it? You mentioned there were some French people at that party.
That was Francois Jacob, and he and I finally got together it wasn't at a party, we were just having a discussion in my room. And
which room was it, do you remember?
It was E5.
Yep.
E5.
In the Gibbs building.
In Gibbs, yes. And that was a very interesting time. And then later, Francois and I went to Caltech to do the experiment and did it in 3 weeks. But just managed only at the last week, because there's also, he describes it in his book, none of the experiments worked. They were very difficult ones, but they just kept on not working until I suddenly realized and stood up at the beach at Malibu, where we were supposed to have a bit of R&R, and said, it's the magnesium. We rushed off to the lab. But that's, it's the magnesium.
It was the magnesium.
It was the magnesium. I suddenly realized that's what's wrong.
The magnesium.
We didn't have enough magnesium. And we rushed off and we set up 3 experiments. And in one of them, the centrifuge failed. But we managed to carry, this thing had to be centrifuged for nearly 2 days. And the centrifuge we were running in had failed, and I had to carry this valuable rotor sort of through the corridor to another centrifuge where we could continue it. And that was quite a test.